Welcome to Brain Matters, the show about all things neuroscience. Whether you're a seasoned academic or just curious about our brain, our guests will be covering everything from cutting-edge research to practical tips for improving our mental health. I'm your host, Thomas von Rijn, and today I'm joined by Dr. Brett Kagan and Dr. Forough Habibollahi, researchers at Cortical Labs, to discuss their work in biocomputing. Brett, Forough, welcome to the show.
Thanks very much, Thomas. Pleasure to be here.
Thank you, Thomas. Pleasure.
So before we dive into the science, could you both start out by telling us a bit about yourselves and the paths that your careers have taken over the years to get where you are now?
Yeah, sure. So I am currently a computational neuroscientist and basically working as a data scientist in Cortical Labs. But how I started was that first I did my undergrad in electrical engineering in Iran. So I was into maths and physics, obviously. But then I was very interested to become a more applied scientist, if you want to say. So I kind of switched paths to do master's in bioengineering or life sciences in Switzerland, EPFL. So basically I took some master's courses in there, and then that's where and how I became very intrigued in studying the brain, the neurons more, and how they're communicating and how they're processing information. And then I moved to Australia to do my PhD in computational neuroscience, and then I was lucky enough to become familiar with Cortical Labs and what they're doing. I think it was in 2020, and then we started working with each other since then, and it's been an amazing journey.
And you, Brett?
Yeah, so I'm Brett Kagan. I'm the Chief Scientific Officer over at Cortical Labs. I've got quite a broad background as well. I started out really interested in the things that make us us. So I started with psychology, and I just found myself more and more drawn to the biological basis of that. And so I moved over, I did some cognitive psychology, some neuropsychology, and then I thought, let's go down into proper biological neuroscience, focused on cellular levels and how that could happen and the pathways in the brain. And then I went and did a PhD in stem cell therapy for brain injury. But the clinical side was very interesting, but it had different sort of troubles and challenges than the ones I was interested in solving. So I was really interested to go back into the basic science research and trained up a bit in bioinformatics, and was always drawn to this idea of these fundamental bases for how life can evolve and become intelligent. And I was fortunate enough in 2019 to be contacted by Hong-Weng Chong, who is the CEO of Cortical Labs, and sort of join up, and we started this kind of crazy idea to see, could you actually harness biological neurons and get any sort of intelligence out of them if they're in the dish? And so now we've been working on that for what feels like my lifetime.
So that was a nice little taster of what's to come. What is Dish Brain, this work that you started over in Cortical Labs? Can you give us an introduction? I think we'll build from there on the science.
Yeah. Well, Dish Brain is actually now defunct. So Dish Brain was a prototype that we did as a basically proof of concept. Is it possible to get any sort of intelligent behavior out of a bunch of neurons you put down onto a dish with little ability to stimulate and receive the electrical signals? We did this work starting from about 2019 to about 2023 and published a few nice papers about it, which we can talk about later. But because the technology has been advancing so quickly, we've actually superseded it and moved beyond that. What it was this idea to see, like, if you encode the right stimulation to cells, could you actually get a meaningful response in a dynamic simulated game world?
So you were growing neurons onto a microarray of electrodes.
Yeah, that's the idea. And we did that in a few different ways. And there's really is the two broad classical ways you can get neurons at the moment, at least legally. One is you can take them from animals, typically when they're still embryonic. So say a mouse when it's been 15 days in gestation. Now, we didn't love that because while animal testing is necessary in some cases, this was a case where we had an alternative. And that alternative was to develop neurons from stem cells. And these stem cells can be generated from just a simple blood donation. You can then use some techniques to turn them into something that's called pluripotent. And you can then take those forward into a range of different types of neurons. And so we started and we wanted to validate, because many people have doubts about whether these neurons that you generate from stem cells are the same as real neurons. So we made sure we compared the animal ones that most people do to the synthetic biology approach that we were adopting. And we saw that actually, amazingly, not only did the human neurons from the stem cell background learn, they actually learned quicker and better than the animal neurons. And so that gave us all the confidence to actually say, great, we don't need to engage in any more animal testing. And that was a great relief for us.
When we had a discussion beforehand, you said that Karl Friston was a collaborator in the research that you were doing. How come that was the case?
I can only narrate the story, I guess, but Brett can correct me if I'm wrong. I believe when the initial idea of Cortical Labs and Dish Brain, it was called previously, when it was proposed, the founders, including Moein Khajehnejad, they did meet with Karl Friston and then they proposed their ideas to him. In the UK.
The thing we were interested in is part of those questions. One of them was like, how or why would a group of neurons do anything? So we basically looked around at a range of different theories, and this was one that seemed really attractive to us. There were clear ways we saw that we could test the theory. It didn't deal with the idea of neurotransmitters or dopamine or anything like that. It dealt with the fundamental properties of information. Particularly information entropy, the amount of disorganization in the system. Now, we weren't actually setting out necessarily to prove or validate that this was true, but what we did think was, well, if we test this really influential and, for that matter, controversial theory, either we find support for it or we find support against it. And as long as the test we've built up has been sort of agreed upon with the founder of the theory, Professor Karl Friston, either way, it's good science, right? We set a hypothesis, we test it, and then it's up to reality to show us what the results were. In the end, we did find very strong support for it, but either way, for us, it was a win-win. We would have discovered something new.
Could you briefly describe what the theory was?
So basically the theory we're talking about, first of all, is called free energy principle that we're all referring to. So what Professor Karl Friston proposed is that any biological system, including our brain, is trying to maintain their stable state in the world despite all the flux of the environment around them. But how they're doing that, the theory proposes that they are trying to actually minimize a metric or a measurement which is called free energy or surprise in this theory. And what this means is that they are trying to minimize the difference between the predictions that they have, by they, I mean biological systems or the brain, the predictions they have from the world around them, and the actual sensations or observations they receive from the world. So the brain kind of wants to minimize this difference, and how it's making these predictions is based on its own internal models of the world, or what we can call the belief system. And this difference can actually be minimized, generally speaking, in two ways: whether the brain can update its internal model or beliefs of the environment to try to match it better with the actual observations or sensations, or it can try to act upon the world and try to change it so that the observations conform to the predictions. And maybe I can give an example here, like from daily life. Let's say you want to drive from home to your workplace, and it's a route that you always take. You know exactly where the traffic lights are, you know the traffic jam, you know how people behave, you know your car and all of that. So you kind of have a really nice model, and you can predict very closely what's going to happen and how you're going to drive to work. But then let's say something happens in a certain day. Maybe there's traffic jam, maybe your car breaks down, maybe there's an accident or something like that. So you kind of get surprised, but you're internally wanting to minimize this surprise. So you want to update your internal model to match the actual things that you're observing. So you can have different solutions for that. Maybe you check the traffic at home and then say, okay, I have an alternative route. I can take the train, which is much more predictable to me. I have taken it before. I know when it's going to leave. I know when it's going to arrive. Or if this pass has traffic, you take another pass that you also know very well. You know what your speed is going to be. So you're all basically always trying to minimize this surprise element for yourself. And if I want to link that to our system, if it was true for the case of our biological neuronal networks as well, which I'm going to call BNNs, this would mean that we can actually influence them. And these neurons, if they're trying to minimize their free energy, they have this internal model of the world around them as well. They have this belief system. And they will learn via updating this belief system or acting upon the environment, trying to change it. And for this reason, we try to influence their behavior by giving them very unpredictable and surprising feedback after they did something incorrectly or made a mistake or an undesirable action in our game environment. So this means they started to learn and adapt by avoiding actions that will cause a lot of surprise to them. And this is basically our closed-loop feedback system that trains the neurons to do a specific task and can open up a lot of possibilities into, finding or making intelligent systems that are as adaptable as actual living organisms.
Thank you. You talked about how one sources cells. How does one develop them, fix them to these arrays, and then you're working with biology, so how do you keep them alive?
Yeah, great question. What we can do is we can develop them, and there's two main ways with the synthetic biology pathway we can go. The first tries to recreate the developmental pathways that would happen, say, in utero. And this method will provide small signaling molecules, and they'll provide them at the right timings, at the right amounts that will tell the cells, hey, turn into a brain cell, now turn into a cortical cell, now expand and grow. So we can provide that. And fortunately, this wasn't a technology we had to invent. There's been nearly 2 decades now of solid work just on this process by labs using these cells for all sorts of things, from just studying development to doing stem cell transplants, as I was trying to do in my PhD. So the other method that we can do that's been a bit more recent is a genetic modification approach. And so you can either use a virus or something like CRISPR and actually get particular genes to be overexpressed in those cells. And now this will give you a neuron that is quite active quite quickly and very stably and reproducibly, but it's not going to capture all the variability you have in more representative biology. So there's pros and cons to both. One takes a lot longer, far more tricky to get it right and consistent, but it gives you something more natural at the end. The other one's far more stable, but you don't get the same diversity and function that you might get otherwise.
So I think you touched on this briefly, Forough, during your description of the free energy principle. But how did you go about training the neurons to play in a game environment? How did they learn?
We realized the neurons basically don't like the surprise, right? Don't like the unpredictable outcomes that they receive when they do something wrong. But then to maybe expand on it a bit more, what we have in our system is that we do have a special type of an electronic chip, which we call multi-electrode arrays, which has a lot of electrodes on it. But then these neurons are placed, a layer of actual living neurons are placed on this chip, and then they start to grow and connect with each other. And you could say they actually make their own design, their own neuronal network on the chip. And then we can interact with them. We can stimulate them using the electrodes, but we can also observe and record their responses using the same electrodes that we have. And then what we did is that we have this designed simulated game environment, an arcade game like Pong in our first works. And what we do is that we feed the information about the game state to these neurons via the simulation electrodes. For example, we can say how far the ball is, how fast it's coming by using different amplitudes or different frequencies of the signal. And then whenever they do something wrong, or basically when we're observing their responses, they're moving the paddle to the wrong direction, we give them this unpredictable feedback, which they showed us they don't like. So this means after so many repetitions of the game episodes, they try to avoid those actions. And this is how they're starting to learn which actions are better, meaning that what type of activity is moving our hypothetical paddle in the game environment in the right direction. And that's how they started to learn and be trained in this game.
So the carrot is nice, regular input, and the stick is irregular, random stimulation.
You could say so. That's what we found in our experiments.
Akin to the Tamagotchi, will people have to feed these neural cells like a pet? Look after them?
Well, it depends on how they do it. But we've actually built up a perfusion circuit system. So that's like a life support system. So it'll actually provide them the nutrients they need. It'll keep them at the right temperature. It'll filter out waste. So it'll preserve them and it'll make it easy. Who's to say where it'll go? I do think that for most people, they'll be using them on the cloud. So people like us who will be running these neural data centers will take care of the maintenance.
Now, in the introduction, I talked about cutting-edge research. Being at the forefront of such a developing industry as synthetic biocomputing, have you had to engage in considerations about ethics and similar practices going forward?
Yeah, we're actually been engaged in ethics all the way from the start. Our first work as a company was actually ethical commentary, not scientific. It didn't attract as much attention, but it was laying the groundwork for trying to do this in the right way. I mean, even the language we use is a matter of contention for some people.
Yeah, true. But like we were actually just discussing with Brett whether we need to generate new language to be able to describe our systems, or just keep it a bit more, simple, accessible, and use a language that already exists, like using words such as belief or sentience. And we have had a lot of discussions among ourselves, but also with the broader community, whether this is the right language to use. So we have been dealing with a lot of these and ethics from the beginning, as Brett mentioned.
And there's some brilliant bioethicists and philosophers out there who are really eager to engage in really good faith in this work. In fact, I found most of them, when they see that the scientists are willing to engage, the ethicists have been really willing to engage. And we've had some great turnarounds. We've had ethicists who have started out being pretty pessimistic about the whole idea of in vitro cells in a dish, really worried about that. And we've been able to turn around and talk with them and say, well, look, here are the benefits. Here are the guardrails. Here's how we can do it right. And, one of the biggest, I think, personal successes like in my career is the fact that we've taken some people who have been really, really concerned about this, who have now gone, well, actually, no, now I can see that there are benefits as well.
In line with the open communication that you said about fostering, can you name some specific concerns that came up that you have since put in either guardrails or tried to address?
Yeah, well, I think there's kind of three core areas that people get really concerned about. The first one is like, how do you source the cells? And we talked about that a bit before, right? And with that, you do get a funny quirk where some people assume that something from a human origin somehow is a human or has some human trait like a soul. And that's actually not matched up with most sort of religious perspectives don't necessarily view that either with what we're talking about. No heartbeat, no tissue, doesn't have a form of anything to represent that. Often it's people like who aren't so familiar in those areas who kind of jump to this reaction. And that's fine. It's an understandable reaction. But at the end of the day, when you get down to it, we're talking about kind of less cells or roughly the same number of cells as a bee. And so then you sort of have to say to people, well, they're just cells. Maybe there's some small differences in how their structures are. They might give them some small advantage in how they process that information, but they don't have a human level intelligence. So are you saying you've never swatted a fly before? And most people say, oh, no, I guess I don't mind swatting flies, and that concern goes away. The second one are the applications, right? Like, what are you going to use this new technology for, right? There's a lot of concern now with AI safety, and it's not necessarily misplaced concern for AI safety. But when you come to a biological situation, well, it's so much more controllable. It's contained. It sits in a dish. It can't self-replicate. It consumes very little power. There's all these benefits that are, for let's say more traditional AI, that are risks, and for us are benefits. And then finally, you have the most tricky aspect, which is the moral status. Could you potentially have a morally relevant trait? And by that I mean, often people use the word consciousness. Could it have a feeling? Could it suffer? And again, this is a tricky one because I can't prove to you that I'm conscious, nor you can prove it to me. There's no clinical marker that's ever been found to show what consciousness really is. And so we don't dismiss this, but we do have to say we have to be sensible about how we approach it. And first off, as I said, back to the language, we need to define what that word means, because there is no definition of consciousness. There's no one definition of sentience. There's a lot of different ones, and people love arguing about it. but none of them actually are like locked down.
Okay, I think we've covered a lot. So I'd like to pause and ask you some light-hearted icebreaker questions, just to give people more of a glimpse at the person behind the scientists. Does that sound okay?
Sure.
Sure.
I'll ask a question and you can both give your answers. We're in Melbourne, the hub of coffee in Australia, so they say. What is your go-to coffee orders?
Dark hot chocolate. Blasphemy, they say.
Sounds good, actually.
Yeah. I do a coffee out of need, but I've never been convinced that coffee is the most tasty beverage that exists out of the whole world.
So I wouldn't be the first shocker. I don't like coffee. I only like the coffee smell. So I would go hot chocolate or tea.
Hot chocolate is very popular in the Cortical Labs break room, apparently.
Apparently.
I think you might have just picked the two people, actually. The amount of coffee we
I
go through
think
is a testament to, I think, the amount of work people putting in. But you might have picked the two people here who don't.
Outliers.
What's the go-to takeaway order? At the end of a long day, no dinner at home.
Pad Thai noodles.
Thai food in Melbourne's pretty solid. Yeah, for sure.
Love that.
Yeah, I would say takeaway order. Yeah, Thai's a pretty good takeaway order as well. Go a red curry. Otherwise, like I'm always a fan of like a pita and good falafel if you can get that. That's always a handy sort of snack on the go at the end of the day.
Brunswick's good for that.
Yeah, that it is.
If you needed half the sleep you do now, what would you do with that extra time?
I would read a lot, not just science.
Fiction?
But
Non-fiction?
maybe fiction, maybe drama. But yeah, I don't get enough chances to read, I guess, as much as I would like to. Yeah.
I'll probably try and take up some sort of... look, to be honest, I'd probably just spend it on work.
Oh, no.
Life is like... But, like, it's funny, when you start out, like, people often talk about a work-life balance, but I think when you pick something up like science, that you're not doing it because it pays that well or it has job security. You especially don't go into a startup investigating, like, leading-edge technologies. You do it because it's a thing that you're passionate about. Like, the work is your life. And to me, that's not a sad thing. It's empowering. I get to wake up every day and do what I love.
Do you both have any hobbies that you spend your free time on the weekends?
Hobbies. I love hiking. Like, just go around somewhere, maybe 1 hour drive from Melbourne. You'll get beautiful hikes there. And I love swimming. Hopefully, if the weather allows.
Nice.
We like, my wife and I like to go sort of, also walks. We have a very energetic cat that we take out with us, so we'll often go take him to the park or go for a small hike with him when he's behaving. And other than that, maintenance of life and house takes up a fair bit of time as well, so
I imagine
quite handy.
I imagine the cat turns some heads.
Yeah, we get some attention on that front. He's quite a static cat, so a lot of people, we almost need to get a QR code on him for the questions. It's always the same questions. Oh, what breed is he? Does he like walks? And it's just like yes. Scan the QR code for your questions.
Okay, I think you've given people a very good introduction to your work and also the world of this emerging biocomputation field. While you have the platform, is there anything you'd both like to promote or plug?
I wanted to say some maybe interesting findings that we've had recently as well, which we released publicly, that we actually did find some more potential to these cells recently in one of our studies, where we saw that they can be more sample efficient compared to the very well-known AI methods in the field of reinforcement learning. So basically, there are these AI methods that are well known to be playing very well in arcade games like Pong, and we have done a very rigorous comparison showing that if we give them the same amount of opportunity, the same number of chances to mess up, miss the ball, and then repeat the game episode again, given the same number of chances, our cells actually are learning faster, so they're more efficient. Now, we're not saying the AI methods eventually don't become pros in the game, which they do, but we're just saying that maybe there is this potential in our cells, like there is in our brains, that they can even be more sample efficient than a lot of the AI methods that we know.
Yeah, I think to wrap up, I would just plug the idea of openness in science, which I think is a theme we've talked a lot about. You know, we're always engaged. So if you're interested, if you're listening to this, don't think like, oh, well, this is just a thing that scientists are doing. I could never join up. I could never do it. Like whatever stage of your career you're at, you know, we've got a bunch of community pages. You can jump on. You can talk to some great people. You can get involved. You can start and look at this. You know, it's going to be a new area. There's growing interest around the world. So come and get involved and see what, you know, we can create together.
If people wanted to reach out to either of you for collaboration, discussion about the topic, where could they do so?
You can go to our website, which is www.corticallabs.com. We also have a Discord and a Reddit. I must admit, I believe people respond to that. I don't have as much time for social media myself. But you can also email info@corticallabs.com.
I was happy to receive messages like via LinkedIn, email. Definitely happy to discuss.
Brett, Forough, thank you so much for your time today.
Thank you very much, Thomas. Pleasure chatting.
Thank you for the invitation. Pleasure, Thomas.
If you enjoyed this discussion and want to learn more about the brain, why not check out our website at brainmatters.nl, where you can find easy-to-follow articles in both English and Dutch to satisfy your curiosity. I would also like to thank the State Library of Victoria for providing us with the space to record the episode. Until next time.